So I'll just make a start. My name's Nyquist Condon, and I'm the Light Microscopy Officer and CZI Imaging Scientist in the IMB Microscopy facility up on level 6. The point of this talk was to just give a really brief introduction to image analysis and how that might, some of the techniques and tips and tricks that people who are coming into the imaging facility, either as new honours students or PhD students, or people who've done microscopy before and just wanted a refresher. So it's a very high level overview, and we have a number of workshops planned throughout the year, and this will give a foundational knowledge to that. Why would we want to perform any kind of image analysis? Well, it's more than just a pretty picture what you should be getting off the microscopes. The images that you get can be both qualitative, so you can get a really nice picture that might be good for the cover or just a qualitative thing, but you can also get quantitative data with meaningful numbers and statistics out of your microscopy data, provided that you do the right thing. And so hopefully this talk will address some of the key concepts for doing the right thing with image analysis. And often we're seeing a lot of researchers come to us saying, publications requesting that we have to have quantification, and so knowing what to do is really important. Image analysis does not equal image beautification. So when we do things in Fiji or any of the commercial programs that we have, we're not just trying to make the image look pretty. It's not Photoshop. You shouldn't be using Photoshop for any of your microscopy image processing or analysis. An image is just an array of numbers at its simplicity that represent the intensity that was detected by the digital capture device. So in this example of a nuclei, if we keep zooming in, we'll be able to see that there's just discrete pixels that have gray values that correlate to these numbers. And a pixel is just a picture element. So that's the 2D representation. But in when we're doing 3D visualization especially, and when we capture data on the microscopes, there is volume information with each slice that we take, and that we refer to the 3D pixel as a voxel or volume element. And in a classic fluorescence microscope image, if we look at it in like its numbers form in a spreadsheet, you can see that all these background values are quite low, and that's the black values, and the object in the foreground, the nuclei, will have these higher values. And there's two main types of images that we'll take in the on the microscopes. There's RGB color images, and these are good for capturing the histology and bright field images, and we capture them with color cameras. Or we capture in grayscale or monochrome for our fluorescence. Now we can have multiple fluorescence images that we all take as 3 or 4 or however many channels of monochrome, and then combine them to see multiple colors, but this isn't an RGB color image, it's different. So when we talk about capturing our grayscale or monochromatic images, they have a bit depth. And this is like a color resolution, or the gray levels of the image. Most of our systems will by default capture in at least 8-bit. There are higher methods at 12 and 16-bit, and these are better for quantification. And one important thing to note about gray values or bit depths is that they have to be absolute. There can't be negative values. So if you have a 1-bit image, it's 2 to the power of 1, and you have 2 options, black or white. For 2-bit, you would have 4 options, and 3-bit, 8 options, right through to the ones that we're more realistically capturing, which isn't in these discrete bins. It's 256 levels of gray, 4096 for 12-bit, or 65,536 for 16-bit images. When we take our monochrome images, we don't have to display them in this grayscale. We can have different lookup tables, and these are just how we map the color onto those intensity values that we've recorded. So if you have a dark background and light objects in the foreground, and you're capturing an 8-bit, you would see that this gradient goes from black being at the background through to pure white, which would have a value of 255, or some derivative in between. We can invert the lookup table so that we can say, that our background is actually 0 and our objects in the foreground are dark. And that can be useful for displaying really fine structures because we can see dark things on a light background a little bit better than light things on a dark background. And we can map colors to them. So if we have a green channel image and we want to show like our GFP as intensity of green, pure green would be mapped to 255 back down to 0 for the background or the dark bits. Now, when we apply these lookup tables, it's important to think about color blindness and how it's displayed can be, may not be what you see, what other people see. So if you have a red sensitivity for color blindness, where you'd have your classic red, green, and blue image, then people might find that hard to see because it would appear like this. Whereas if we modify the red channel and display it as magenta, then this is how it would appear to people who are colorblind. And we can still see these differences where they're lost in this image. And one of the main programs that I use, Fiji, has a really nice tool to simulate color blindness. And you can have a look at all the different ones and you can see how the images will appear to people with different color blindness. I will admit that in this talk, some of the slides are red, green, and blue, and I haven't updated them. So I apologize in advance to anyone with that. When we have our microscope images, it's important to think about how they have dimensions. So we don't just have to have a 2D Polaroid picture. We're capturing lots of different dimensions that can be the width, the height, the depth. If we're capturing 3D, we can capture multiple channels and we can capture multiple time points. Usually they're designated by these nomenclature. So X and Y, Z for depth, C for channel, and T for time. And we can have lots of different types of dimensions of data. So you could just have a single color 2D image that's represented as an XY image. If you had multiple colors, then that is a 3D image. You've got 3 dimensions. But you could also have single color over time, or 3D with multiple colors, which would make 4D, or you can have multiple colors in 3D over time, making 5D. So to simplify things, we usually only refer to 2D or multiple dimensional, because all of these additional multi-dimensional images are treated the same way by most softwares. And it's important to note that if you capture on different microscopes, the order that it stores the data within the files, or if you want to extract down all the information into the 2D lowest form, the outputs might vary because they don't all capture in the same order. One vendor might have channel as a higher priority than depth. So just keep that in mind. You can also, something else that you have to keep in mind is the pixel size. And this is a property that you define when you acquire your data. More pixels over the same area can help us to see objects in a better way, but it doesn't mean having the most amount of pixels will give you the most resolution. So if we have quite a large pixel size of, say, for example, 2 microns, we wouldn't be able to see all of the structures. And as we move to a smaller pixel size, we can generally see the image and the detail a bit clearer. But just having smaller pixels doesn't always mean that you'll have more resolution. There are limits to this. And throwing more pixels at an image doesn't always mean that you'll have more resolution. But using tools in the software like Optimal or auto setting the light path for the right size of the image, the right pixel size and zoom can get you on the right track to having the right settings for your experiment. Now, when we talk about resolution, it's a physical property of the optical system or the microscope. And there are multiple different ways to talk about resolution, but one of the main ways is to be able to discern 2 objects from one another. So if we have this high resolution example and this low resolution example, then we can see that as we zoom right in, that if we have high resolution, we can detect the 3 parts to each buckle, whereas in the low resolution, we can't resolve that. Now, simply having more magnification doesn't always mean that you're going to have more resolution. So we have multiple different microscopes that I listed before, and we have lots of different objectives on them. If there was the highest resolution option available for everything, there would just be one objective. But we have lots of different types because they all have their own specialty and special use. When you are looking at an objective and deciding which one you may want to use, the optical resolution isn't determined by magnification. It's determined by the NA and the wavelength. So I should say NA, sorry. And the way that we calculate the theoretical resolution is using the Rayleigh formula. So the Rayleigh formula is 0.61 times the wavelength of the emission over the numerical aperture. So for example, if we had GFP signal that we wanted to capture on a 1.4 NA objective, then our formula would look like this, and it would calculate down to being 224 nanometers, which is about the optical limits of most microscopes. Another important thing to think about is image brightness is determined by a different formula. It's formed by the wavelength over the numerical aperture to the power of 4 divided by the magnification squared. So this would mean if we had a 63 times 1.4 NA objective and 100 times 1.4 NA objective, then the 100 times is about 40% dimmer. So as we zoom in magnification and expand that area out onto our camera, it's less photons coming from that area. So moving up in magnification usually means that you'll have a dimmer image at the camera. Now, there's a lot of other things that can affect it, and this is quite a simplification, but the immersion media and your sample refractive index can affect things like the resolution and brightness. Bleaching of your sample can affect resolution and brightness, and how thick and deep that you're going in the sample. So come and talk to us, and we have objectives that are better designed for short working distance, long working distances, different refractive indexes. So come and talk to us is the key point. Now, in a biological context, resolution can mean using a 20 times 0.8 NA objective. We could see some detail of these nucleoli, but if we move up to the higher resolution objective, we can clearly see that there's multiple structures here. And so choosing what you need to see and what, how big that would be, or how big you would anticipate that object to be versus what objectives to use is really important to think about from the very beginning. Once that you know that you can see two objects in your image, or one object if it's only that, you want to ensure that the objects themselves stand out above the background. And we use signal to noise, or the signal to noise ratio, to describe how bright an object is, the signal, versus the noise, or just the background in our image. Having a higher ratio is better. So high SNR means that we can quite easily segment out our object from the foreground versus the background. And it's usually required to resolve these objects. So if we have a very nice example in the middle here and we drew a line across one of these beads, we would see that it has relatively low background with quite high in the foreground. If we have high foreground and high background, then that ratio isn't very much, and it's not very big, and it'll be hard to segment these objects. So while it might appear obvious to us where they are, mathematically determining them, it can be difficult. And likewise, if you have really low background but still really low signal, then you have that same smaller ratio and you'll struggle to do segmentation and some of the analysis. So it's important to have the object being bright, but you don't want to go too bright either, because if you saturate your images, then you lose information. So when you overexpose your image, you're saturating it. So in the case of these three objects here, we can see that the left object has a value of around 100. The center object, it's saturated across the peak and it's maxing out at that 255. And we don't know what's happening here. Is this just like a normal Gaussian peak, or is there information in here that we've lost because we've saturated it? You can't differentiate any structure within that one saturated region. And it's important to plan for future results. So not just the image that you're capturing, so think about the controls. So if you have a time course where you have cells that are taking up a sample, it's probably going to get brighter. So I wouldn't set up your imaging parameters on, time point 1 minute. So think about controls in microscopy images. And one way to look at the intensity to know where your data is sitting across this whole, 255 or 16-bit range is to use the image histograms. So a histogram is just showing that the pixel intensities are representative across this range, either by a logarithmic scale or just by summing the number or the frequency of them. And so where you have an object that sits above background, then you can safely know that you should be able to segment because you can see this dip. So black pixels, or really dark with a low value, are going to be all stacked up here on the left side, while the objects that the bulk of the data here, it's skewed across this range from about maybe 25 to 150. I can also see from this histogram, there's a little bit of a spike at the end here, which would suggest that some pixels have been saturated. But the main thing to think about with your histograms when you see them is, the foreground objects and the background and where they sit, and if there's a bit of a clear curve. So I've taken a screenshot in the Zen program where there isn't really an obvious distinction between the background and the foreground. So it's in the red channel, at least here, we see it's just kind of degrading down. And so it's almost like a linear gradient from having lots of dark pixels to, some light pixels. But there wouldn't be a clear distinction like you can see in this histogram. So it's important when you're capturing to look at this data to think about the future and what steps down the track you might be looking at doing. It's really important as well to be consistent with your acquisition settings. So when you have two samples and you want to compare them, it's really important to go through the whole experiment treating them the same. So you have to be really consistent. So whether it's exposure time, if you've got a camera-based system, or the gain on a PMT-based or confocal system, they should be the same. And this applies to every setting. So you should have the same cover slip thickness, the same antibody labeling times, the same concentrations of antibody. On the microscope, you should have the same lamp intensity, the same laser power, the same pinhole. So when you can compare something and you say, like here in sample A, there's lots of mitochondria and they're really bright. And in sample B, there isn't many mitochondria and it's dim. So maybe our treatment has affected that. You don't want to just up the exposure and see, oh, look, there's still red signal there, because you can't compare them anymore when you've changed the setting between things. And as I mentioned before, think about your experiment, not just the image that you're looking at, but where it sits in the context of things. So your imaging settings like laser power, exposure, all should be set on the brightest and the dimmest samples. So not to have it too low that you can't detect anything at time 0, but not too high that it saturates by time 0.5 and you've got 3 more ones after that that will get brighter. So do you expect them to get brighter? Do you expect them to get thicker, or your tissue to move deeper if you're looking at like a 3D section? All these things you should take into account and think about that, it's going to change and how that will affect your imaging parameters. Another key thing in microscopy is our sampling frequency. So we use a method called the Nyquist theory, which is basically we should capture it 2.3 times the event or expected event size. So because we're converting like an analog representation of a natural event into a digital representation. So if we're taking a picture of something, it's going to be binned into pixels. So we want to make sure that we have the right pixel size. If we have an object that's moving over time, then we want to make sure that when one cell moves from the first position to the second, that we've captured enough frames to know with confidence that it moved straight there? Or did it, crawl away and another cell crawl in and it looked like the same cell? So when we have our microscopes, we have tools that can help, make sure that we're sampling at the same rate. So if we have a cell that's about 10 microns, you want to make sure that your pixels are not, 10 microns, because when you have two cells nearby, you won't be able to differentiate them. And this could also be endosomes if you're looking at things and you want to really zoom in, or if you're looking at a fish, you want to make sure that your vessels are, a certain distance apart and that you have that sampled at a rate that you're not just going to have one pixel representing the whole vessel. And if you're looking at temporally resolved structures like a cell crawling around, and to make sure that you're capturing it at the right frequency so that with confidence that that's the same cell, or what you're trying to describe is true. So how do you capture a good image? Well, you use the tools that the vendors have provided you. Usually they're pretty good. You can use something like range indicator. So range indicator works by changing the lookup table. So instead of it's just like red objects or objects in the foreground are red and backgrounds black, and it's hard to tell, is that 255 saturated red, or is it kind of 250 and am I okay? So if you change to, in the Zeiss case, the lookup table where you have red as a maximum value and blue as a 0 value, and everything else is gray in between, you'll quickly see that any red pixel here is saturated. And maybe you should think about adjusting your settings so that across your whole experiment, with all the different treatments and conditions, that you don't have these saturated pixels, because saturated pixels mean that we've lost resolution. We've lost the ability to, resolve those structures. And in the case of Leica, they use a different color mapping, but it's the same principle. If it's a 0 value, it's shown as green. And if it's a saturated value, it's shown as blue. And they have this color gradient between these two values. Now you can do this after the fact in Fiji or image analysis programs. They have these so that you can look at it and see, oh, I've oversaturated some objects, but you can't go back and change it after the fact. So try to think about using these tools on the microscope. Use tools like Optimal. So when you're setting up a confocal acquisition, Zeiss gives you this button right under where it says the pixel size of your image. Set it to optimal so that it will say, for the objective you've chosen and the wavelengths that you've chosen, how many pixels do you need and what size should they be for your capture? And if you're using the Zeiss system, there's a button here in their top left corner that does the same thing. And plan for your experiment. Talk to us before you, capture 50 slides of data and, it ends up being all junk if you've done the wrong thing and you can't do any quantification. So think about, are you using the right objective? Maybe the microscope doesn't have the perfect objective for you. So talk to us, because we have lone objectives that we can swap out and put on that are more specialist for different things. If you're capturing Z steps for a 3D, data set, are you capturing enough Z steps? So often we see, people come to us with image analysis questions, and it's 512 by 512, which is the default, and it's a 1 micron step size, which is the default. It means that you can't, know for sure that the objects that you're seeing, you haven't missed out endosomes, or you haven't, got too much of a gap in your data, and you can't with confidence say what, you are seeing in the image is true. Likewise, have you got enough time points? Is your sample going to get dimmer or brighter or change over time? And have you considered that? And what are your controls for your images? So, you can talk to us if you have questions about your controls for your experiments. Sample preparation is really important. If you have junk or crap going in, you will not get anything better than crap coming out. So use the right cover slips or the right dish or the right glass for your experiment. So you should be using number 1.5 cover slips. So this is what every objective that we have in this building is designed for. So they're corrected for a thickness of 160 to 190 microns. You can get cheaper cover slips that are outside that range, but know that it's going to affect the quality of the images you get. If you wanted to do STED or TURF, for example, you have to use these. You just cannot achieve the resolutions possible without using those right cover slips. The store downstairs stocks cover slips in this size, and most of the commercial dishes you buy from like MatTech or Nunk should have a 1.5 cover slip option. Use modern dyes and fluorophores, so Alexa dyes, Addo dyes, Star, M Emerald, M Neon Green. These will be brighter and have better photo stability. Again, in some of our methods like STED, you really need specific labels for that method to work. Clean the slides and the cover slips with ethanol before you image. They are really dusty in this building, and dust tends to be autofluorescent. So you see these big bright things, and if you've got that perfect cell that has the right example and there's a massive bit of crud next to it and a big crystal of PBS or something that's autofluorescing, it can be quite, frustrating. And your slides should be sealed with clear nail polish and using a reputable mounting media. Imaging fixed slides should be done pretty promptly. They fade over time, and depending on that mounting media you've used, it may or may not contain preservatives, and then your dye or label might have just faded, and you can't then know for sure that your treatment made it with juice, or unless you've got the right controls as well. And if you're doing imaging, a live imaging experiment, think about the right conditions for the right imaging media. So is it buffered? Ideally, it should be phenol red free because that's autofluorescent and can cause problems. Again, talk to us about your experiment, and we can point out some things that you might not have thought about that you should be planning and factoring in. And you should know what file format or file type to save in. Different formats exist for different things, and you really need to know which ones that you should be using. JPEG is a lossy format, and you really need to avoid using it. The compression algorithms in the JPEG file format mean that you lose information like resolution or bit depth. Ideally, you should be using something like a TIFF or an OME TIFF that's lossless, and it's a good common format, but you might still lose some metadata information. The vendor-specific file is always the best because it will have all the relevant metadata. So the vendors all tend to have their own proprietary formats. So they produce a file that's bioformats compatible because bioformats is a third-party group who write all of the metadata explaining, and so that in the Zeiss file, they might call magnification a different term to a Leica format, or how many channels and all that information. So all these different microscopy vendor-specific files should be openable in Fiji as long as your bioformats is up to date. Often they have a free software available to open, but it's quite limited. They'll happily sell you a $10,000 offline license to run the software. So we have most of the offline softwares that you can use. The free versions are quite limited. Otherwise, you should be using something like ImageJ Fiji to do that. There's only one or two formats that we've come across in the facility that are not openable in free software like Fiji because they are from licensed software. I've spoken briefly about the resolution limits and things like that. There are ways that we can break the optical resolution limit with some of our microscopes. So as I said at the beginning, there's some other things that can be affected. So not only is adding more pixels, there are some methods around breaking this resolution. So usually, depending on the wavelength, a microscope's diffraction limited to about 220, 240 nanometers. But there are methods called super resolution methods that overcome this, such as STED, and this uses an additional laser to deplete light around the excitation area, and you can get down to 30 nanometers resolution. AirieScan on two of our confocals uses a special detector array to get over that resolution limit and down to about 120 nanometers. We also have a TURF microscope, which isn't strictly super resolution, but it limits excitation in Z to being around 100 nanometers. So you can see right at the cover slip or right at the cell surface receptors and things like that. There are other methods, Palm and Storm, that use special dyes and statistical analysis blinking to get beyond that resolution limit. And we have STED, ARICN, and TURF in IMB. We also have SIM on one of our systems, the Lattice. So you can, if you're interested in some of these methods, talk to us. There's also, not only is there, the basics of what you can do with setting up your image around pixel sizes and things like that, there are best practices and conformities in the field that you need to apply to. So there's the NMRC Code of Conduct, which is a document that sets out and prescribes, what we should be doing as researchers. As you can see, it's a strong research culture will demonstrate honesty and integrity. Researchers must foster and maintain research environment of intellectual honesty and integrity. Must tell the truth, and publications should be complete and report negative findings. And researchers must take all the steps to ensure their findings are accurate and properly reported. So that's a very general overview. There is also a scientific publication or peer-reviewed publication where they outline, experts in the field have outlined what is the basic rules or expectations of people handling microscopy data. And these include just acknowledging that digital images can be compromised in inappropriate manipulations. So straight off the bat, knowing that you can do something inappropriate with an image is important. Any manipulation should only be performed on a copy. So you should always have a backup of the original file that's safe and unchanged. Simply adjusting something on the whole image is usually acceptable. So making the whole image brighter or thresholding on the whole image or something is, again, acceptable. Cropping is usually acceptable. If you're going to compare two images, they should be captured under identical settings, and all the acquisition processing should be identical. Manipulating one area of an image and not on another is pretty questionable. Using software filters to improve image quality is not recommended, so that, not Photoshop. Cloning or copying objects into a digital image from one part or another image into the same image is very questionable. It's the polite way of saying, do not do that. Intensity measurements should be performed on uniformly processed data. The data should be calibrated to a known standard. So usually we're not ever going to use intensity data to say that there was X number of molecules or X copies of things. There's too much variation, but we can compare amongst our standards or our samples to say this one's brighter than the control, but we can't say that there's 20 extra copies of GFP there. Avoid those lossy compression file types, and magnification and resolution are important, but they are not the same thing. And be careful when changing the size in pixels of a digital image. There's also some examples of image manipulation. So here's an example where they have selectively enhanced this labeling of this object. So they've made only this object brighter, and that's, against the rules. They really liked this cell here and this group down here because we can see that they've added it to the image. So usually you can't replicate the backgrounds. So when you saturate up an image that's presented in a figure, if you see something like this, it's highly likely that that has come from a different image. Most publications have tools that check for these things automatically when you upload your publications now. So this is from a paper in 2014, where now, many years from that, the technology's come along the way. But it doesn't mean this stuff doesn't happen. This is an example from UQ, where an image has been manipulated. So it's a gel, and when you enhance it, you can see that a band has been rubbed down here. And in another example from UQ, this histology image is what was presented, but this was the image that was actually captured. And all of this damage had been stamped and holes filled in with other regions of the image, and this these nodules were added and replicated. They're all the same. So this is usually pretty career-ending stuff if you get caught. It's, you know, not something that we ever want to see coming out of IMB or UQ. And there are people who publicly look at this information, and one of them on Twitter is Elizabeth Bick, and she has quite the eye for forensic changes to images. And this was one of her tweets from a little while ago now, but showing from that's what was in the publication to these are all the same objects that she's matched, and they've basically built up these images. So you can get caught. It's pretty, you know, there are tools or there are people who are really good at this. So it's not something that you ever should be doing. Lastly, I want to spend a couple of slides talking about your data storage. So we should all be adhering to the FAIR data principles and data storage principles. So FAIR stands for findable, accessible, interoperable, and reusable. So your data should be perhaps have a DOI generated against it, or it should have all the accompanying metadata so that it's findable, so that you know information about the image. It's not just a JPEG saying control, because that is meaningless to anyone else. It should be accessible, so it should be open and shareable, and people should be able to understand what you've done. So whether that's through the settings in the metadata, through the naming conventions of what you've done, what you've labeled it with, that's where the accessible is. Interoperable is community agreed formats, language and vocabularies. So there are standard metadata. So having conformity to bioformats addresses that part of the FAIR data principle. And reusable. So data can be licensed to other people. It should be maintained at levels and have all the provenance information about what you've done to that data so that someone down the track can know what's happened and they may need to use it. So one thing to think about for the FAIR data principles is to develop a naming convention. And you should be consistent, especially if you're just starting honors, starting your PhD. Don't call something control and treatment. That's meaningless. Name it like fluorophore-1 protein, so GFP and your protein, Alexa 488 and your, antibody that you've targeted it to, and then like a number sequence. When you put all this information in a folder, it should be written in the format of year, month, day, so that then it becomes sorting. It's really easy. You don't want, if you're doing a PhD over multiple years, you don't want all your stuff from January and at the top, and then all your stuff from February below that. You want it the other way so that over the years, it all sorts out nicely. You should be using Research Data Manager, mainly because it ensures that your data is accurate, authentic, reliable, complete, that it's all there. It retains its integrity and the reproducibility of your research. It's secure, and it minimizes the risk of data loss. I have a box under my desk of dead hard drives. They break quite frequently, and that's not from physical abuse. It's just they fail, and you should make sure that everything is on a backed up device. So don't store it on your laptop. It might get stolen. It might get left on the train. It might get coffee spilt on it. Have it backed up and stored somewhere. It's available in accordance to both the ARC Code for Responsible Conduct of Research and the FAIR guiding principles, and it meets the requirements of all the funders, journals, and regulatory bodies. Everything, all your data, from the original acquisition to anything that you've done post-processing and your quantification on it, and if you've done 3D visualizations, should all be stored on an RDM cache. There's really no excuse now to not be using it. With that, thank you. And if there's any questions, I'm happy to answer them, or if you have really in-depth questions, we can have a chat after the fact in person.